Hello my dear students, this is Dr. Pooja, your biochemistry faculty. Well, now in this video we are going to start up with a very new and an interesting topic, proteins and the amino acids. Proteins and amino acids. So now the thing is that what are proteins? What is their biomedical significance? What are amino acids? What is their significance? So if we talk about proteins, these proteins are actually the polymers of the amino acids. So proteins are simply polymers of amino acids. Right. Then this term protein, actually it is derived from the word proteus, which means holding first place, which means holding first place, and this was coined by Berzelius. Right. Now the question is, Berzelius coined this term. If we talk about the biomedical significance of proteins, proteins are a very, very important source of energy which help in building up of the muscle mass. So this is what is the significance also. So if we talk about the proteins, they are useful in building up of the muscle mass. Up of the muscle mass. Now, if we talk about proteins, there are so many important examples. Just say if I talk about enzymes, we know enzymes are the ones which are catalyzing the biochemical reactions. Enzymes are all proteins, obviously except ribozymes, but majority of the enzymes are proteins, except ribozyme, right? Then if we talk about the immunoglobulins or the antibodies, antibodies are also proteins. We know what are antibodies, which protect us from the antigens. Then if we talk about defensive proteins, antibodies are there. If we talk about transport proteins, like albumin is there, then you can say hemoglobin is there. These are all the transport proteins which help in the transportation of so many important things. If we talk about albumin, albumin is the carrier of drugs, albumin helps in the transportation of bilirubin, and hemoglobin is the respiratory pigment. So this is also protein. If we talk about the hormones, there are so many peptide hormones which are present, like insulin is there, glucagon is there, they all come under this category. Oxytocin, vasopressin, angiotensin, everything will come under this. So you can just imagine that proteins are having such important functions to perform in the body. Then if we talk about this, the most important function of a protein is that proteins are having maximum specific dynamic action. They have maximum SDA. SDA means specific dynamic action. Very important. Right? So these are, or they are assimilated, giving more energy to the body. So these are some of the important functions of the proteins. Now the question is, protein is the one, what amino acid is producing it? So whatever properties that protein is getting, it is from the amino acids. So basically we have to concentrate on these amino acids first, because they will be the one which will be required to form the proteins or the polypeptides. Now what are these amino acids? Now these amino acids are actually the compounds which have alpha carbon atom which is associated to both two functional groups. So amino acids are basically C-alpha, or which have a alpha carbon atom, right, carrying two functional groups, right, means they can accept a proton, they can donate a proton also, and this is the generalized structure of a amino acid, NH2COOHR. Okay, this becomes the generalized structure where R will be having different classification, right. Now, if we talk about this becomes the general structure, so I'll just highlight it like this. Now, there are 20 standard amino acids. There are 20 standard amino acids. Okay, now the question is, these 20 standard amino acids are classified into different types. Okay, and the most important thing which we have to focus here is that these 20 standard amino acids are the ones which will undergo metabolism, means they will be synthesized, they will be degraded, they are going to form specialized products, and they are the holy solely sources which are going to expel this amino group in the form of urea. Okay, so we can say that proteins are the only macromolecules. This is a very important property about proteins, that proteins are the only macromolecules which removes this amino group as urea, because we are ureotelic organisms. Okay, so proteins are the only macromolecules which remove this amino group in the form of urea, or we can say that they are the ones which is giving rise to nitrogen balance. So this nitrogen balance which exists in the body, it is due to the proteins. Very important. Okay, now coming over to this, how these 20 standard amino acids are classified, this is very important. So this is what we are going to focus now. So whenever we talk about the classification, first way of classifying is as per R group. So whenever we have to differentiate, or whenever we have to classify the amino acids as per R group, and always remember this will be the generalized structure. Now this R group is having different ways. So if we talk about classification as per R group, first is the aliphatic amino acids. Aliphatic amino acids, right. So here you can say aliphatic mein what all will be the example? Aliphatic. The examples would be glycine, alanine, and branched chain amino acids. And branched chain amino acids include BCAA, they include leucine, isoleucine, and valine. Right. So if we talk about the aliphatic amino acids, or if we talk about glycine, glycine is having this type of structure. Everything is same. Now you will see that NH2COOH, here instead of R, H will be there. So this becomes the structure of glycine. Now when this H is replaced by CH3, NH2COOH, CH3, this becomes alanine. Okay, so aliphatic amino acids will include glycine, alanine and branch chain amino acids. Glycine is having this structure, alanine is having this structure, we can say CH3 is there. Okay, so R group will change, and if we talk about the branch chain amino acids will have this type of structure, you can see, right? So valine, leucine and isoleucine, they all will come under branched chain amino acids. So all these are aliphatic amino acids. Then second is when this R group is replaced by, say, acidic group. So acidic amino acids will be 2. An example is aspartate and glutamate. Right, now why they are acidic amino acids? Because they have an extra COOH group. They have an extra COOH group, and aspartate is converted into its amide derivative as asparagine, and glutamate is converted into glutamine. So basically they are the amides which are produced from aspartate and glutamine, right? Then third will include basic amino acids. Now basic amino acids, जैसे acidic are there which have an extra COH group, the basic amino acids will have extra amino group. Right, and the example of basic amino acids is, the mnemonic is HAL, H-A-L. So it is histidine, A for arginine, H-A-L, L for lysine. So we can just remember the mnemonic as HAL. Now when we talk about this, the difference between this is that histidine has a characteristic imidazolone ring, arginine has a characteristic guanido group, and lysine has amino group at the epsilon position. So histidine has an extra imidazolone ring, right? Arginine has a guanido group or guanidium group, and lysine has at the epsilon position an extra amino group. And if we talk about this structure, let me see if the structure is there. So we can say that this becomes the structure of, as you can see that, okay. So here if we see these structures, this is a characteristic imidazolone ring. So this structure is of histidine. And here guanido group is there. So this becomes arginine. So this becomes the structure of arginine and this becomes histidine, right? Then we come over to the fourth one. What aliphatic amino acids we have done, acidic amino acids we have done, basic amino acids we have done. Then fourth is hydroxyl group containing amino acids. Containing amino acids. Now if we talk about hydroxyl group containing, there are only 3 examples, serine and threonine, right? Then we come over to the next category which involves sulfur containing amino acids. Sulfur containing amino acids, and these sulfur containing amino acids include cysteine and methionine. Structures we have already discussed when we have discussed we are going to discuss in the metabolism, because metabolism, understanding of metabolism is perfect when we see the structures. So here even if you don't remember the structures, metabolism mein they are clearly mentioned. Cysteine and methionine mein if we see the difference, this is the generalized structure of an amino acid, because I told you only R group is different. So here it will be having CH2SH means this is sulfhydryl group. Sulfhydryl group is there. On the other hand, methionine, generalized structure, but the only thing is that it has CH2SCH3. Yani ki it does not have a free sulfhydryl group, it has methyl group along with this. And that is why methionine is required for transmethylation. So this is sulfur containing amino acids. Then we come over to the sixth one. Simple, we can say that they are the amino acids. Now first of all, why they are known as the amino acids? Because they do not resemble the structure of amino acids. But yes, their properties are similar to that, so that is why they come under this. And the example of amino acids is proline and hydroxyproline or hydroxylproline. And if we talk about the structure of proline, proline has this type of structure. And if you talk about the structure, you can simply say that in proline, this is the alpha carbon atom. So this alpha carbon atom is not free to form a stable peptide bond with any of the amino acids. Okay, so this becomes the structure of proline, which is an amino acid, not an amino acid. So this way of classifying the amino acids is as per their R group. Then second way of classification is as per the nutritional requirement. Per the nutritional requirement. Now when we talk about as per the nutritional requirement, they are classified into three subclasses. Or what are these three subclasses? Essential amino acids, semi-essential amino acids, and non-essential amino acids. Non-essential. Right. Now, if we talk about the definition, we can simply say that essential amino acids are those amino acids which cannot be synthesized in the body and they have to be provided from outside diet. Semi-essential are obviously required by the growing children, or you can say lactating mothers or pregnant ladies in bulk. And non-essential can be easily synthesized in the body. Now, how to remember essential, how to remember semi-essential? So, out of those 20 standard amino acids, always remember 8 are essential, 2 are semi-essential and rest of the 10 are non-essential. Again, there is a mnemonic to remember them. TV till 8 p.m. TV till 8 p.m. where T stands for threonine, V stands for valine, this T stands for tryptophan, this I stands for isoleucine, this L stands for leucine, lysine, phenylalanine and methionine. Right. Now here I just forgot one more thing to classify these aromatic amino acids, where we wrote aliphatic, no? So 7th category I can say they are the aromatic amino acids. Okay. And these aromatic amino acids include phenylalanine, right, tyrosine and tryptophan. And this tryptophan has an extra indole ring present in it. So we can say that tryptophan can come under heterocyclic amino acids also. So ideally these are the 20 standard amino acids. Okay, let's go back to essential ones. So essential mein we can say TV till 8 p.m. is the mnemonic. for these essential amino acids. Then coming over to semi-essential, there are only 2 semi-essential amino acids because they are required by the growing children and they are required by the lactating mothers or the pregnant mothers. Lactating and semi-essential will include arginine and histidine. And always remember, if we have to compare arginine and histidine, this histidine can come under essential amino acids. But arginine is generally semi-essential. Okay. So this classification is based on the nutritional requirement. Then third way of classifying these amino acids is as per their metabolic fate. As per their metabolic. So as per their metabolic fate, we classify these amino acids again into 3 classes. One class is glucogenic, second is ketogenic, and third class will represent both glucogenic and ketogenic. Right. Now, what are glucogenic amino acids? Always remember glucogenic, as the word suggests, which gives rise to glucose. So we can say which forms glucose. If we talk about ketogenic, as the word suggests, keto means they will give rise to some keto group. So they form या give rise to acetyl coenzyme A. Glucogenic and ketogenic means they can give rise to both. Now coming over to the examples of this, glucogenic and ketogenic में you can remember a mnemonic as PITT, P-I-T-T, where P stands for phenylalanine, I stands for isoleucine, this T will stand for tyrosine, and this T will stand for tryptophan. So PITT will come under both. They are glucogenic as well as ketogenic. If we talk about ketogenic, there are 2 amino acids which are strictly ketogenic, leucine and lysine. So leucine is a potent ketogenic amino acid, right? Lysine is partly glucogenic also, but predominantly it is ketogenic. That is why it comes under ketogenic amino acids. Okay. Then comes glucogenic. So you can say 2 in this, 1, 2, 3, 4 in this, and rest of them are glucogenic. Now under glucogenic amino acids, the most important, the most potent one is alanine. Rest are, you can say, glycine is there, serine is there, all will come under etc. Come under glucogenic. Okay. So this is the classification as per their metabolic fate, very, very important. And then we come over to the last part of classification of this, classification as per the charges. Now, if we talk about as per the charges, always remember we can classify them into polar and non-polar. Polar and non-polar. Now, this polar can be charged, it can be uncharged. Can be charged, it can be uncharged. Right? So if we talk about the charged ones, always remember, example we have to remember about ABC. ABC means it will include acidic amino acids and basic amino acids. We know acidic are aspartic acid and glutamic acid, and basic are lysine, arginine and histidine. Uncharged mein it is going to include cysteine, but it is polar. Okay, then we can say non-polar. Non-polar will include methionine and tryptophan, or you can say tyrosine. Tyrosine or phenylalanine, they all will come under non-polar amino acids. Okay? So this is classification as per their charges. So as per the charges, polar and non-polar, the polar can be further differentiated into charged and uncharged, and non-polar will be like mostly the aromatic amino acids will be non-polar. Here we can classify tyrosine also, but tyrosine is having an hydroxyl group at the para position, because if we talk about phenylalanine, phenylalanine is strictly having a aromatic structure. Okay, tyrosine is actually para hydroxyphenylalanine, so hydroxyl group is there which might give it a little bit of polar character. So it is not strictly non-polar, right? So this is the classification as per the charges. And this is how we classify the amino acids. Let's quickly see how we have differentiated them. We can say that as per R group, because this will be the generalized structure of all the 20 standard amino acids, and this R group will be changed. So this R group can be aliphatic, so they will come under aliphatic amino acids. Examples are glycine, alanine and branch chain amino acids. Second is acidic amino acids, which will include aspartate and glutamate. Basic amino acids, which will include histidine, arginine and lysine. To remember this, we can just remember the mnemonic as HAL. Okay. Then OH group or hydroxyl group containing serine and threonine. And if we talk about sulfur containing, examples are cysteine and methionine. And then comes aromatic amino acids. They will include phenylalanine, tyrosine and tryptophan, and then will come amino acids, which will include proline and hydroxyproline. So this is as per the R group. Then as per the nutritional requirement, amino acids are classified into essential, semi-essential and non-essential. So if we talk about essential, the mnemonic to remember them is TV till 8 p.m. and these are essential ones. Semi-essential are only two, arginine and histidine, but when we have to compare the essential nature, compare between these two, histidine is given more preference. Then comes non-essential, so we can say the rest of the amino acids except this will come under non-essential. And then we come over to the most important classification as per their metabolic fate. So if as per metabolic fate, glucogenic, ketogenic and they include glucogenic as well as ketogenic amino acids. To again remember this, I told you, you have to just remember a mnemonic PITT. Phenylalanine, isoleucine, tyrosine and tryptophan, which will come under both. If we talk about only ketogenic, leucine and lysine. Now leucine and lysine mein I told you ki lysine is actually predominantly ketogenic, partly it is glucogenic. So since it is predominantly ketogenic, so it has to come under ketogenic ones. And glucogenic mein rest will be the ones, but alanine being the most potent ketogenic amino acid, right? And then as per the charges, polar is there, non-polar is there. So we can say all the aromatic ones, because benzene is non-polar, they will come under this. Methionine, it has sulfidyl group but attached to CH3 hydrocarbon, so it also becomes a non-polar ones. And if we talk about polar, acidic and obviously basic amino acids will come under polar. Okay. Now, this is the way we classify these amino acids. Now, after these 20 standard amino acids, there are two more additional amino acids, which is known as 21st amino acid. Now, what is the name of this 21st amino acid first of all? It is known as selenocysteine. Right. Now, why it is referred to as a 21st amino acid? The reason being this is that selenocysteine is actually coded by a nonsense codon. It is coded by nonsense codon, right? And that nonsense codon is UGA. Why? Because it does not have a corresponding anticodon and it is synthesized by a process of co-translation. So, process is co-translation. Now, the question is that what is the significance of selenocysteine? Ek to it is synthesized from serine. It is synthesized in the body from serine, and the name of the enzyme is selenocysteine ATP synthetase. ATP is required. Our significance is it is a coenzyme for so many reactions, or enzyme, coenzyme for so many enzymes, like the first and the foremost enzyme is glutathione peroxidase. Right. Second is thioredoxin reductase. Third is selenocysteine P and deiodinase. Right. So, we can say that selenocysteine, even though it is coded by a nonsense codon, it is referred to as a 21st amino acid, but since it is a coenzyme for so many important enzymes, glutathione peroxidase is a very important enzyme as it helps in maintaining the integrity of the cell membrane of RBCs. Thioredoxin reductase, DNTPs, help in the synthesis of DNTPs or the conversion of NTPs to DNTPs. This is related with iodine metabolism, and selenocysteine P is also required in the gene synthesis. So, these are the important enzymes which require selenocysteine, but it is coded by UGA, that is why it has been given a separate name, 21st amino acid, right. Then there is 22nd amino acid also. Now, this 22nd amino acid is known as pyrrolysine. And again it is also coded by a nonsense codon, and the codon is UAG. It is coded by UAG, and again the process is co-translation. So there are, I can say, 20 standard amino acids and 21st and 22nd amino acid also. Okay. Now, after this, then we come over to what are the properties of these amino acids. Now, the most important property of these amino acids, first of all, is isomerism. Second property is the isoelectric point. These are the two very important properties of the amino acids. Now, first of all, if we talk about isomerism, so if we talk about the isomerism, the most important isomerism which exists in the amino acids is stereoisomerism. Stereoisomerism, right? Now what happens here is, we know if we compare, because what is the reference molecule first of all? So we will take the structure of glycine first. So this is the structure of glycine. NH2, COOH, H. So this glycine, right, if it is going to form a mirror image, okay, the mirror image formed by glycine will be this. That means it lacks a chiral carbon atom. As you can see, this is not a chiral carbon atom, because for a molecule to exhibit stereoisomerism, chiral carbon atom is must. Chiral carbon atom should be there. Right? Now, if we talk about glycine, glycine lacks a chiral carbon atom. Okay, or it has asymmetric carbon atom. Right? And third important property about glycine is that since it lacks a chiral carbon atom or asymmetric carbon atom, it is optically inactive. And since it is optically inactive, it is going to form superimposable mirror images. Well, mirror images. Right? Then the second amino acid is alanine. So if we talk about alanine, alanine is having this type of structure: CH, NH2, COOH and CH3 here. Now if we have to produce a mirror image of alanine, it is going to form this type of mirror image: NH2, COOH, H and CH3. Now if we talk about this, this is a chiral carbon atom and this becomes alanine. So I can say that this alanine has chiral carbon atom, chiral carbon atom. Right? Second important point about this is that it is going to form non-superimposable mirror image. So it is optically active. So second point is it is optically active and it can form non-superimposable mirror image. Non-superimposable mirror image. Now the question is, if it can form a non-superimposable mirror image, what are the main isomers? Because it is going to form the stereoisomers. So if NH2 group is on the right hand side, we call it as D-alanine. And if NH2 group is on the left side, we call it as L-alanine. Right? So we can say that NH2 group on the right side is known as D-alanine and NH2 group on the left hand side is known as L-alanine. Now if we talk about the digestion in our body, digestion and absorption में L-alanine is more easily absorbed, or you can say L-amino acid are easily absorbed in the body, easily absorbed and assimilated. Okay, but there are certain exceptions also, like D-aspartate and D-serine. These are the two amino acids which are in D conformation and they are present in the brain. Right. So we can say that if we talk about the isomerism here, simply we can say that alanine has a chiral carbon atom, it is optically active, Because it is going to form the non-superimposable mirror images here. And how do we define them? We can say that if NH2 group is on the right hand side, it will become D-alanine, and if it is on the left hand side, it will become L-alanine. And L-amino acids are more easily absorbed and assimilated. Okay, but there are certain exceptions. So D-aspartate and D-serine, which are produced in the brain, they are having D-conformation. So isomerism, if we talk about, so it is capital D and capital L enantiomers, right? And the reference molecule here is, if we talk about the reference molecule is alanine. As it was glyceraldehyde in carbohydrates. Okay, so this is the first property. Then second property, which is the most important property, is isoelectric point. Now these amino acids have a tendency to occur as a Zwitterion. Jैसे if I write the structure of generalized structure, I can say that amino acid is having this type of structure. Okay, so if we have, because it is having an amino group, it is having a carboxylic group, and this is the alpha carbon atom. So what happens is, it can actually donate a proton and it can accept a proton, right? So I can say that it can donate a proton or it can accept a proton. So this amino acid, or those substances which can accept a proton, which can donate a proton, are known as amphoteric substances, and this type of ionic structure is known as a Zwitterion structure. So I can say that amino acids can occur in Zwitterion, and all the amino acids are amphoteric substances. Amphoteric substances means they can behave as an acid and a base. Now the question is that we have to separate the amino acids from a polypeptide, and since amino acids are carrying both the charges, they are having a positive charge, they are having a negative charge, we will let them to move. A stage will come when there will not be any net migration of these amino acids. So that particular point, when there is no net migration of an amino acid, will be known as isoelectric point. Very, very important. So how do we define an isoelectric point? Isoelectric point is a point, is defined as a point when there is no net migration of an amino acid. Okay. Or we can say that the positive charge will be equivalent to the negative charge, or we can say that net charge will be 0, or we can say that the amino acid becomes neutral. So we call this as isoelectric point, and it is denoted by Pi. Very, very important. Okay, and this becomes the basis of separation of this also. So just try to understand that all amino acids, I told you, they are having a similar structure. The only difference is in the R group. So they are having an amino group, they are having a carboxylic group. That means they can donate a proton, they can accept a proton, and that is what is an acid is. Acid which can donate a proton, base which can accept a proton. And those molecules which are having both, they are known as amphoteric substances. So all amino acids will be amphoteric in nature, but they are existing as Zwitterion. So a stage will come whenever they have to migrate. But when the migration stops, or we can say the number of negative charge is equivalent to the positive charge, or the net charge on that amino acid will be 0. That becomes the separation of that amino acid from the mixture. So this is known as isoelectric point. And always remember, at isoelectric point, the net charge and amino acid are neutral. Okay. Now the question is, suppose if I say the pH is less than pi, there can be only two conditions. pH can be less than pi or pH can be more than pi. pi is isoelectric point. If pH is less than pi, जैसे in the stomach, so, and if pH is more, that means towards alkalinity. So what will happen is, whenever pH is less, it has to be positively charged. And if pH is more than pi, it has to be negatively charged. Negatively charged, right? So positively charged, protonated. Negatively charged, deprotonated. And the common example of this is albumin. Now this albumin, pi is 4.7. Okay, so now you see the pH of pi of albumin, isoelectric point is 4.7. So whenever we say albumin in circulation, is negatively charged. Now why albumin is negatively charged in circulation? Because pH of blood is 7.4. So pH is more. So here, if pH is more than pi, it is negatively charged, right? That is why albumin in circulation is negatively charged. On the contrary, this albumin, when it is in the stomach, so albumin in the stomach is positively charged. Now why it is positively charged? Because there pepsin is there, and we know that stomach has acidic pH. Acidic pH, that means pH is less than pi. So this decides that how the molecule moves and how everything is going on. Okay, so this is what is pi. And suppose if we talk about, if we have to make a simple titration curve, and if I talk about for a single amino acid, right, suppose this is the curve, here pH is there and here alkali is added. Okay, and starting mein it is ionized and here it is unionized, right? Here it is ionized and here it will be partially ionized. Right, and if we compare it with 2, so what will happen is, for a double titration curve, it is going to form this type of structure, like this, 1 and 2. So here this is 1, this is 2. This is again, it is unionized and here it becomes completely ionized. And here we take again the alkali which will be added, and here it will be the pH. So overall we can say that it will be equivalent to pKa1 plus pKa2 divided by 2. So this becomes pKa1 and this becomes pKa2. So this is how we actually define this pi. And this is how, for a double, you can say, titration curve, it will be like this. Initially it will be totally unionized. Let it move. So it will stop at one place, it will stop at another place, and then it will be completely ionized. And that is what is pi. So if we talk about pi, what is the characteristic feature of the molecules at pi? So always remember, at isoelectric point, there is net charge, so obviously it is 0. Net charge is 0, but maximum precipitability is there. Ability is there, okay. Then minimum solubility is there. Solubility is there, and least buffering capacity. Least buffering capacity will be there, right? And that is why we always say, and it is calculated by Henderson and Hasselbalch equation. So Henderson and Hasselbalch equation, we can simply say that pKa is equal to plus log of base over an acid. And we always say histidine, whose pi is roughly 6.7, which is closer to blood, whereas it is 7.4. So histidine can have good capacity, and histidine is required for the buffers, and moreover histidine, we can say that it is a very active, or it is a very stable amino acid at a physiological pH. Okay. So these are the two very important properties of amino acids. One is isomerism and the second is your isoelectric point. So this is all about this video. So in this video we have discussed about the classification and the properties. In the next video, what we are going to discuss is, we are going to study that how these amino acids are forming the proteins and how these proteins are aligned to form the stable polypeptides. So that's all in this video. Thank you so much.